Okay, in this screencast, we're going to be talking about viruses. All right, viruses are pretty funky little particles. Their structure is basically pretty simple. It's just DNA or RNA surrounded by a protein coat. The protein coat is called a capsid. Now you need to make sure you remember capsid versus capsule, which is the outside of a bacteria. Now, in this diagram below, we're taking a look at a particular kind of virus called a bacteriophage. Phage means virus, and this is a kind of virus that attacks bacteria. The reason that we're going to look at the bacteriophage in a little bit more detail is just that the way in which it infects bacteria is a little bit easier than the way in which a human virus attacks human cells. So to make sure we understand how viruses work, we're going to keep it a little bit simple. So the virus the bacteriophage virus has DNA and RNA. It's surrounded by a protein coat. It's got a tail and some tail fibers. Now, viruses contain DNA or RNA. They have proteins and they reproduce. So, are viruses alive? Well, science at this time has decided the answer is no. There is no kingdom of life that includes viruses. Viruses have no nucleus, no cytoplasm, no organelles or cell membrane. They cannot carry out cell functions, and they only reproduce when an infected cell builds new viruses. If you look at the data table below, they don't grow, they don't maintain homeostasis, and they have no metabolism. If science decided at some point to redefine life, it is possible that viruses would be considered alive, but then we would have to have a kingdom of life specifically for viruses. All right, so viruses are not alive. What do they do? How do they work to get cells to reproduce them, to replicate them? Well, the process of replication of a virus is called the Lysogenic cycle. And this is where we're showing you a bacteriophage. That's the little red thing that almost looks like a spider sitting on a blue box. The blue rounded box is a bacteria. We're showing you this because it's simpler than what would happen actually in a human cell. So step number one, you see the spider, the virus, sitting on the bacteria. So the virus attaches to the host cell. In step number two, the virus injects its genetic material. In a human cell, it would actually enter the entire cell, but for a bacteria, it's just going to inject its DNA. Step number three, the host cell's DNA is degraded. So notice the green DNA is breaking apart. That belongs in the bacteria. It's falling apart. Number four, the host cell is forced to put together new virus parts. That pink DNA in the diagram, all that DNA says is how to build new viruses. And every cell, whether it's a bacteria or one of your cells, our cells do what the DNA tells them to do. And if they've been injected with DNA that says build viruses, the cell says, okay, I'm going to build some viruses. And it starts building virus parts. Now notice in step four that it builds them and builds them and builds them until the cell is bursting with virus cell parts. And eventually in step number five, the cell breaks open. We call that lysing, where that's part of the name Lysogenic cycle. When it lyses open, the cell breaks open and these built viruses come tumbling out. And now any one of those viruses can move on to infect another cell. Now there is another cycle for viruses that's called the lysogenic cycle. The lysogenic cycle is when viruses remain inactive within host cells for days, months, or years. It's basically a time delay for the lytic cycle. This is what happens. The viral DNA is inserted directly into the host's DNA. And then when the host's DNA is copied during replication, the viral DNA is copied along with it. So initially you have one cell that's infected. Well, once the cell goes through mitosis, you now have two cells that are infected with the viral DNA. Well, that DNA remains dormant for sometimes generations isn't the right word, sometimes for decades, that's the word I'm looking for, until there's some triggering event that causes the virus DNA to reactivate and it enters back into the lytic cycle, which is what we learned on the previous page. The triggering events are a bit of a mystery to science. We know that sometimes stress in a person's life can cause a virus that is inactive to become active, but apparently ultraviolet light can also do that. Chemicals, especially some chemicals in some medications, heat, and sometimes we just don't even understand why. There's some reason that a dormant virus suddenly becomes active. An example of a dormant virus is the shingles virus. It's the same virus that causes chickenpox, only in some people it remains dormant in their DNA until some triggering event causes them to develop the shingles, a case of shingles. Now, there's different types of viruses, and I'm not going to spend a whole lot of time talking about them. The DNA viruses are the most stable viruses. They have DNA, they inject their DNA, the host cell copies the DNA and makes RNA, and then the RNA tells the cell how to build more viral proteins. Some viruses have RNA in them. The virus inserts its RNA, the RNA moves directly to the ribosomes, and the ribosomes build the viral proteins. Where it becomes particularly difficult are the retroviruses, and we'll get to that next. The most famous example of a retrovirus is HIV. What happens with a retrovirus is the virus has RNA, but then it has a particular enzyme called reverse transcriptase. And the reverse transcriptase makes DNA. It goes backwards. Instead of going out to the cell, going out to the ribosomes and making a virus, it goes backwards and makes DNA. And the reason that the retrovirus is such a problem for science is that this reverse transcriptase enzyme makes gajillions of mistakes. And in the process, it creates mutations for that particular virus. So that makes it particularly difficult to create vaccines. HIV has obviously been very, very difficult for science, and we, as of the date of this screencast, have not come up with an effective vaccine for vaccine, and it's largely due to this reverse transcriptase creating so many mutations, so many variations of the HIV molecule. Let's take a few minutes right now just to explain exactly how HIV works. First of all, what happens is when a person is infected and when the HIV virus makes it into the body, the virus stops or docks at a receptor, a very specific receptor on a macrophage. The receptor is called a CD4 receptor. The macrophage has another receptor called CCR5. It is the combination of the two receptors, CD4 and CCR5, that allows the HIV to enter into the macrophage. Now, T cells also have that initial receptor called CD4, but they don't have the CCR5 receptor. So it's like a 2-series lock, and they only have one lock, and I don't know if that explains it very well, but initially, HIV can only enter the macrophages and is not able to enter the T cells. Once it's inside the macrophages, HIV's enzyme called reverse transcriptase, we talked about it earlier, reads the virus's RNA and starts to create virus DNA. But this reverse transcriptase makes many mistakes, and that leads to many mutations of the HIV viruses. Eventually, the HIV viruses made by the macrophage are released. But this is important. They're released by the process of exocytosis. If you remember from our Lysogenic cycle earlier, we said that the cell typically lyses open. It bursts the cell open. Well, the macrophage is able to release the virus harmlessly through exocytosis. So this means that initially the macrophages are still in the body and the immune system isn't damaged initially. However, over time, because reverse transcriptase keeps making all these mutations, sooner or later the mutant HIV will eventually require a new coreceptor. Instead of the CCR5 coreceptor, these mutant versions now require a different receptor called CXCR4. The only cells that have the CD4 receptor and the CXCR4 coreceptor are the T cells. So now HIV starts infecting the T cells. The problem is that when HIV infects the T cell, starts to divide, and then leaves the T cell, instead of leaving harmlessly through exocytosis, it does what viruses typically do, which is bursting out of the cell membrane, and it destroys the T cell in the process. When the T cell count begins to drop down, we actually identify that as a person having AIDS then. When the immune system is no longer functioning correctly, the person moves from the category of just HIV infected to actually having developed AIDS. All right, treatment and prevention of viruses. Unfortunately, to date at the screencast, we have almost no ways to treat viruses. There are a few antiviral drugs. One you may have heard of is called Tamiflu. These antiviral drugs interfere with viral DNA and RNA synthesis, but at this point they are not particularly effective. So we rely on vaccines. There are two different kinds of vaccines that are subtly different. The first kind is called an inactivated vaccine. In that case, the virus is quote unquote dead. Of course, we don't really believe that the viruses are alive anyway, but anyway, and the viruses cannot replicate. The other type of vaccine is called an attenuated virus or attenuated vaccine. In that case, the virus has been genetically altered. It is still functional, but it doesn't cause the disease. Your immune system fights off these diseases that have been injected into you, and you develop a long-term immunity, an active immunity. Attenuated vaccines tend to be more effective vaccines than the inactivated. And that's it for viruses.